Introducing this video brings mixed feelings to me because of the crazy times we're living in and how it's affecting everyone. My perspective as a graduate student of paleontology that is trying to finish my degree on time, well, it certainly has complicated every facet. But it also resulted in strong foundations with which to start this channel. And I'd like to thank everyone for watching this video, those who are watching live, and all the people that have been supporting this channel. The online thesis defense was challenging, but a successful endeavor from all parties involved. The entire event took over 2 hours, but I cut down the public-facing version to under 30 minutes. It includes the presentation of my thesis and some examples from the Q&A session afterwards. I will link the full unedited version in the description below for those that are interested, but this version captures the point of the research without compromising the critically examined scientific findings. So if you have any additional questions, feel free to ask in the comments. And finally, hit the like and subscribe button if you like this and the other videos on my channel. My next video will likely be something lighter and more entertainment focused to bring a little levity to these times. Now, on with the online thesis defense. All right, well, yeah, again, thanks to, a big thank you to everyone that is joining us for my thesis presentation about ontogenetic and adult shape variation in the endocast of Tapirus and its implications for T. polkensis from the gray fossil site. Now, I want to break down what an endocast actually is and what it represents for mammals so that we can understand how useful they can be for paleontology. So a naturally formed endocast happens when a skull gets buried in sediment, fills in the space that used to be occupied by the brain tissue, and eventually, after so much time, that sediment infill will actually harden, fossilize, and cast the brain out of the brain case, which serves as the mold. Now, that's a naturally formed endocast, but nowadays these are often created digitally with the technology that I'll talk more about later. And for mammals, endocasts are really good proxies for the brain shape, the brain size, because of the really tight fit of the mammalian neural tissue within that brain case. There's not a whole lot of non-neural tissue that occupies that space in mammals. Now, early paleontologists recognized how useful endocasts can be. Some of the earliest work was actually done on horse brains and horse endocasts. Evolution of the Brain and Intelligence is an important work. It's still popularly cited, basically controlling for body size and talking about how big, different animals' brains are. Data was taken from mammals, birds, these large groups of vertebrates. Body size was controlled for in order to talk about, well, okay, this animal's brain might be bigger, but is it bigger than expected given how big the animal's body size is? Very, very important. Now, we know the endocast is a really great approximation of the size and shape, the external appearance of the brain. But recently, it's been verified quantitatively for birds at least that when you break down the different regions of the brain as they appear on the surface of an endocast, comparing that surface area is a viable way of comparing the volume ratios of all of those different brain regions. And then other internal structures also scale with overall brain size. What researchers found is that when the overall brain size gets bigger and when the volume of gray matter gets bigger, this white matter that's internal to the brain, which kind of serves as the internal wiring, the connectors between neurons, that gets disproportionately bigger. So the implication there is that the bigger the brain is and the more gray matter you have in the brain, the greater volume of white matter that you need. And that's really important because what seems to be popping up is the volumetric changes between the different regions of the brains and their correlations to animal ecology and animal behavior. The principle of proper mass is a kind of a general tool used to suggest, or used to deduce that the bigger a specific region of the brain is for any given animal, the greater the importance of that region's function is in the ecology of that animal. So if the, olfactory, regions are disproportionately bigger, well, olfactory areas, that means like smelling information, that means we interpret that as saying that that's important in the ecology of that animal. And the same would go for auditory, visual information, because fortunately, the organization of all of those regions are very conserved throughout vertebrates, even if the shapes are different. So an example of that is in this graphic here, where we have a more basal brain and its neurosensory processing areas illustrated, and then a derived mammalian brain. And along with the locomotor ecology and sensory ecology, they also include like phylogeny studies with this too. But I'm more interested in the sensory and behavioral ecology applications for Tapirus and Tapirus polkensis. Social and sensory ecology for Tapirus is basically solitary, largely nocturnal, maybe sometimes crepuscular, and very little evidence for conspecific cooperation. Relationships don't really extend in the greater lifespan of the animal. And this map down here, scientists were collecting DNA from tapir droppings in their natural habitat and using that to put a spatial component on related individuals and their territories. And what they found was that if closely related tapirs are not any more likely to overlap in their territory than more distantly related individuals. They don't aggregate in large groups, and they don't aggregate in small groups that are cooperative and have long-term relationships. As far as paleontology, a single species death assemblage in paleontology is often used to interpret social behaviors in extinct animals, but only if the geology fits a certain criteria. This image up here is a shot of the ashfall fossil beds, which is an example of how you can have a large number of individuals at various ontogeny stages, and even if they're sexual dimorphic, you can use that to investigate what their social system was like and what their mating systems might have been like, because we can be confident that they all were living and dying in that place all at the same time. Now, the Great Fossil Site has a lot of Tapirus polkensis individuals, easily over 100 at this point, if not even more than that. But the geology doesn't quite meet the criteria to be able to say that, yes, we're seeing a different social ecology pattern for Tapirus polkensis. And that was one of the things I was most intrigued about, because endocranial studies is another method of researching and testing for these social and behavioral ecologies in extinct animals. So that prompted this endocranial endocast investigation in Tapirus polkensis and extant tapirs. So this sort of research leans heavily on computed tomography, which is a X-ray-based scanning method that basically sends X-rays through solid objects, and then by their density of the solid object is able to render a series of 2D images that computers can compile into one large 3D model that you can work with. And I used ORS Dragonfly to segment the endocranial space. In the case of the Tapirus polkensis infant, there was some digital compositing necessary to complete the endocranial shape. We found ontogenetically similar specimens of those missing bones and 3D Builder to compile the surface scans of those into the CT scan data, and I used that to kind of guide the segmentation of the endocranial space for the infant. And then I also collected scans that were done in previous research studies. I classified them into infant, subadult, or full adult age categories, along with extinct and extant. Now, in order to test if Tapirus polkensis varies from Tapirus alone, we had to take this data and create a brain body size allometry to be used to predict expected brain size based on the body mass of the different individuals in my study. All of this goes into calculating something called an encephalization quotient, which is a neat little index to rate how far above or below the observed brain mass is based on the expectation from its body size. So this resulted in a brain body size equation following the methods of Berger et al. And Body et al. It is based on adult extant tapirs, because from that you're able to plug in the T polkensis body size estimate and see what the expected brain mass is. Tapir's polkensis endocast encephalization quotient is actually the highest out of all of them in the data set. But when I calculated the z-score for it to see if it counts as an outlier, it's not. It's on the upper end of it, but the z-score doesn't imply an outlier. The coefficient of variation is also 6.36, pretty low. Now the morphological description, I'm going to talk about just a couple of the things that I found most interesting. For me, the separated anterior telencephalon, and by telencephalon, I mean to say this anterior portion of the brain here. Within that space, in dorsal view, you can see the separation of the olfactory bulbs and the frontal lobe areas, and then additionally 5 other identifiable lobes of the forebrain. The pituitary gland, which is where a lot of hormone regulation happens within the brain. This is signified by the hypophyseal fossa, and that's pretty well developed on this adult endocast of Tapirus polkensis. And then the nerves left some pretty good impressions. The cerebellum, or the hindbrain, so this region right here, is largely comprised of a natural endocast signified in yellow. The subadult was a little different, much more rounded and bulbous, heavily concreted, and just overall not as good of a quality. An endocast, the surfaces are a little too smooth for me to confidently identify the different lobes. The olfactory bulbs here are more medially constricted, and they don't extend as far out anteriorly. The anterior telencephalon is not fused as in the adult. And then finally, the infant endocast in my study, going back to a much more clean endocast here. It's still more rounded with a more curved dorsal surface, but there's a lot more concretion on that ventral surface, which actually made these ventral structures pretty easily identifiable. Some gyri and sulci are starting to appear, which is the cortical organization, so the folding of that tissue that you think of when you imagine a fresh brain. That appears to have left some impressions on the infant Tapirus endocast, which is pretty cool. The anterior telencephalon is not fused, and those olfactory bulbs are much more ventrally situated in this endocast. The frontal lobe occupies a greater relative portion of the whole telencephalon, and the hindbrain occupies a relatively smaller part. Now, the geometric morphometrics did not turn up very useful results, but a lollipop diagram, which is a way to show the direction of landmark shifts, do show telencephalic elongation. The discriminant function analysis tried to see if the shapes of the endocasts plotted according to classifications of extinct and extant, and they did not. It was a pretty much a statistical failure. And the only promising element that came out of the geometric morphometrics was the canonical variance, which tested for ontogenetic variation. And the adults do plot together, but only within a 0.8 confidence ellipse, which is something that I really don't want to claim as being super statistically significant, especially when the polkensis infant, again, is plotting closer to the Pinchaquai subadult than it is to the Pinchaquai infant. Starting to analyze all that data, well, Tapirus polkensis is a phylogenetic dwarf, which means it's got a smaller body size than the extant living members of the genus. But based on my research, it does not appear that that has any effect over the relative size of its brain. There's elongation of the telencephalon going on. Qualitatively, you can observe that when you compare the Pinchaquai endocast and the indicus endocast, elongate versus anteriorly blunt and more rounded. And also another thing to consider is that shape changes in a brain throughout ontogeny does not always mean volume changes. So we really should contrast enhance some wet specimen Tapirus crania with the brain still intact, or use an MRI to try to really peer into those subsurface neural tissues. Qualitatively, there are some interesting changes going on. That separation of the olfactory bulbs in the front and the anterior telencephalon, increase in the hindbrain relative volume versus the whole brain volume, and the decrease of the telencephalic regions throughout ontogeny. And then in thinking about what that could mean, well, the motor coordination centers developing a greater relative volume throughout ontogeny makes me really speculate on their impact on the control of the musculature in the Tapirus polkensis trunk, that mobile proboscis on their face. So I'd imagine that if those motor coordination centers in the brain are developing and growing throughout ontogeny, then that may impact their ability to be coordinated with their muscles in their trunk as well. But that's something that would also need experimentation on living tapirs. Their sensory ecology, even though early on tapirs were assigned a olfactory dependency, that's not unfounded. Their olfactory bulbs are quite large, and that mobile proboscis, they use it all the time. They sniff. They use sniffing behaviors to explore environments. But I also was able to find a lot of reports about vocalizations being important too. Based on that, and also the sizes of the auditory centers of the brain, I really would not rule out vocalizations as being very important in the overall sensory ecology of tapirs. But again, that would be something that would require living specimens to really experiment on. Finally, that the Tapirus polkensis accumulation is probably not related to any major variation of social ecology and just a preservation or taphonomic bias. On the positive side, is that means that we can actually do zoological studies of modern tapirs and their environments, and basically use that to help inform our knowledge of the gray fossil site ecosystem, because their sensory ecology is likely so conserved. So I'd like to thank some people, and a quick preview of my references and questions. I remember one of my first conversations with Dr. Wallace, I just had me scratching my head because, essentially why here? If these tapirs, behave like modern tapirs, then they really don't interact. And if they, are overlapping their territory, that's not a regular thing. Like that is very rare. They don't, and no more than groups of maybe two. So it just left me scratching my head of wondering, like, how could this largest collection of fossil tapirs in the world have happened in this one spot when tapirs don't historically, interact with each other all that much. So it led me, speculating about, well, maybe they're, hanging around this as a resource, leading to a greater amount of social interactions in a way that cannot be, modeled by the extant variety. I was super excited to render the endocast and take a look at what its brain looked like, because if it had been different in any significant way, then yeah, it would have led to a big revelation about why the tapirs show up in such great numbers here at the gray fossil site, and cast doubt a little bit on the assumption that, a tapir is a tapir and that all tapirs behave the same. That might not have been the case had the endocasts varied their morphologies in any really significant way. A lot of the investigations into the correlations between brain size and then regional volume, brain regional volume ratios, they're based on modern taxa. So they're correlating these brain morphologies to, one of them in particular was habitat type, whether they lived in closed environments, meaning forests, or open, like grassland type habitats, which I thought was awesome. But they didn't include any fossil taxa in that. So considering we have such a shift from closed habitats to open habitats throughout the Cenozoic of North America, I would love to look at horses at large. And then probably the first thing that could test that would be the Teleoceras apisoma from the gray fossil site. Because here we have, a same genus, Teleoceras major that they find out in Nebraska, and Teleoceras apisoma, which is the one that we find here in a more forested environment. The evidence from modern taxa would suggest that those two animals should have different brain sizes associated with the cognitive demands of living in a closed environment versus an open environment. And I think the Teleoceras apisoma skull we have, I've never looked inside of it, but it may produce a pretty awesome endocast. And I'd love to compare that with other rhino endocasts too. So the second part of that question, how can it fit into the broader research? Well, the biologists that study neuroscience, they find all of these relationships, And we can use that to basically form predictions about what we should expect along that deeper macroevolutionary scale. So if the biologists in the modern data that they're collecting really holds up now, it really should hold up in the future. So we really should be taking that modern data source and using that to create and test hypotheses about these fossil endocasts, because that would either support or refute notions about how tightly are those correlations really? Do they hold up through deep evolutionary time? So I think this and other studies of fossil endocasts are important to kind of support the modern biological data sets. I'll start with the most extravagant, I guess, possibility, and then talk about what's more what's the word like a conservative of a hypothesis. And brains can sometimes have lateralization in their processes, which means that one side operates with a different series of synaptic pathways than the other side. But the research on that is looking at the actual geography of the synapse, is not like overall size. And then it also is something that comes up more often when you talk about the primate lineage, where one side, operates very differently. Another, I think the more likely scenario is that, considering these animals are living in a tropical, in a hot climate mostly, it could be a way to reduce reduce the surface area to volume ratio in the brain of this animal, which might help it keep cooler. I know that tends to be a thing with body size, where the larger you are, the greater your volume to surface area is, and you hold on to your heat more. So a large mammal in a hot environment may have problems with overheating in its brain if it doesn't have some way of kind of cheating it and splitting it up into two halves. And the last thing of speculation that I would take that is that when you're looking at animal cognition, and this is sailing into a little bit more of a complex world than my thesis was really targeting, but it's really all about the way that the neurons are able to connect to each other within a region and then across regions as well. So one thing that I would speculate on from the division of the bulbs and the anterior frontal lobes is that it limits the number of inter-region synapses that are able to form, which also would limit the amount of sensory input and motor output possibilities, I guess, because there's only a certain amount of there's a lesser amount of synapses that could possibly form. But again, when you start getting into that, then you get into a really complex array of speculation of animal cognition in an extinct animal that is not quite there yet, as far as being able to talk about in fossil species. I'm not really inclined to believe that nature can select for skull adaptations related to eating and mastication, but those changes resulting in a neuroanatomy that's not characteristically fit to manage all of those processes, because osteological features don't like if we're thinking about it embryologically, neural tissues start forming very, very early on in that prenatal ontogeny. And a lot of other soft tissues also kind of pre-develop before the osteology does. So I really think the answer to that is that it's more of a coevolution thing. There's still merit in saying that, changes to these premises of feeding ecology or social ecology or locomotor ecology can be predicted by brain size. Like, yes, they may be subjected to selection on other things, but that doesn't necessarily mean that the resulting neuromorphology is not also characteristic of, acting as a central processor for the animal's input and output. It's just a different way of looking at it, a different part of the anatomy to look at. Well, thank you for listening. If you made it this far all the way to the end of the video. I was really nervous when I first gave this presentation online, because it's weird when you have to talk live onto the computer and think about all of the people that might be listening to you. It's a little different when you're recording because then you have time to edit it. So throughout the editing process, I realized just how nervous I was, and I tried to confront some of that in the revisions. Now, I linked the original version of it if you want to see the unedited live event, but everything in this video I still am proud of and would love to discuss more in the comments. So thanks again for watching, and have a good week.